Here we go with Chapter 10, Part 2. What we have already covered is chromosomes. We have covered mitosis. And today, in this lecture, we're going to talk about meiosis. So here we go. Firstly, meiosis is involved in sexual reproduction, and mitosis is involved in asexual. So mitosis can be involved in asexual reproduction. Sexual reproduction is meiosis. Mitosis also has a lot of other roles, as we discussed earlier. It is also involved in the replacement of dead cells in the repair of damaged tissues. But it can also be used to simply copy one single-celled organism to two single-celled organisms. That's another thing that mitosis can do. Meiosis can't do that. So before we begin talking in detail about meiosis, I want to just give a general overview of asexual reproduction and sexual reproduction. Firstly, in asexual reproduction, there's just one parent, and the offspring are all going to be identical to the parent, and they are also identical to each other. So everybody's identical. Essentially, we can say that one parent clones itself, and everybody's the same. In sexual reproduction, there's going to be two parents, as we know. Offspring are not identical to the parents or each other. Back to asexual reproduction. The parent reproduces by dividing cells by mitosis. So this could be a single cell, as I said, dividing into two cells. But also there are in rare cases, big animals, especially lizards, tend to be kind of the most common one. And what will happen is the lizards will consist only of females, and these females, in their ovaries will divide by mitosis and then produce eggs. And then they'll produce eggs. So this can also happen in sort of macro life forms, the big ones. So ovaries would just divide by mitosis, produce eggs, and then these would of course produce individuals who are the same as the parent, clones. So what this would ultimately mean is that there would essentially be clone lineages of these individuals out in the population, out in the environment. So each mother would just produce more of herself only, and then there might be another mother who would be producing more of herself only. And you can think of what the consequences of this might be for evolution. There would be no diversity except genetic diversity except that arising from mutation, which is not that much. Okay, so in sexual reproduction, the parent reproduces by dividing their cells by meiosis. And what meiosis does is it divides the chromosomes by half, and then we get these sperm cells plus an egg cell. Then we get back to the full number of chromosomes. So the sperm has half, the egg has half. Half plus half is 1. All right, we're really cruising on our math skills here. Okay. In asexual reproduction, it is most common in single-celled organisms and then also plants. Some of you might be gardeners, and you might know that sometimes with some plants, you can cut off a part of the plant and then just bury it in the dirt, and it will regrow into an entirely new plant. That's a kind of reproduction as well. In sexual reproduction, it's more common in animals. Plants also, of course, do sexual reproduction. Even single-celled organisms like yeast cells, yeast involved in brewing beer, making bread rise, they will also do a form of sexual reproduction as well, but it is most famous in animals. Animals essentially appear to need to do it. Some species have evolved ways of not doing it, but they are much, much less common. And in fact, a lot of plants can do both. Both asexual and sexual reproduction. So the topic for this lecture is meiosis. How is it that we produce cells that just have half the amount of DNA that their parent cells had? before I continue though, a little bit more on why is sexual reproduction most common. So I mentioned in animals that most animals reproduce sexually, and that would be right here, sexual reproduction. And you can see what's going on here. So here's a female, and she has given birth to 2 offspring, 1, 2, and one of them is a male, and the male cannot have offspring itself. So the female, this daughter here, goes on and finds another male, hopefully not the brother. In fact, many mammal species are unable to recognize their siblings, but hopefully she finds something, a male that's not related to her, and reproduces again. And we get a female here and a male. Again, this male cannot reproduce itself, all by itself. And then this daughter, or female offspring, finds another male and produces another female and another male. Again, the male cannot reproduce. Now, this might seem a little bit weird to say, because you could also maybe make the argument males can reproduce. So this male right here, this one, could find a female and reproduce with that female. Yes, that's true. However, let's take a look at asexual reproduction. What goes on there? The population can grow exponentially. That means really fast. Female gives rise to 2 offspring, and both of them are identical to her, so they are both females. And they don't have to go out and search for a mate, decide which one's the right one. They can simply give birth to more offspring. And both of the offspring can reproduce. So we go from 1 to 2 to 4 to 8 really rapidly. So asexual reproduction is super efficient and fast, which raises the question, why is it that so many animals, most animals, bother to reproduce using sexual reproduction? And the answer is essentially some of the details in meiosis, which we'll address both in this lecture and then a little bit more so in the lab, where I'll get into the details of meiosis a little bit in more depth. So, meiosis. A few things you need to know about it, and I wrote them down here. Your book says them as well, but a little bit more long-winded. Number 1, meiosis involves 2 divisions of the nucleus. Mitosis only involves 1. Second bullet point, reduces the chromosome number by half, or it reduces the amount of DNA by half. It is involved in sexual reproduction only, and the daughter cells are not identical. So meiosis is involved in these fellas: egg cells, sperm cells, and both of these sex cells are the result of meiosis. So some type of cell in the reproductive organs of males and females undergoes meiosis, 2 divisions of the nucleus, and produces sperm if the individual is a male, and produces eggs if the individual is a female. Reduction of the DNA by half. So the sperm has got half the normal amount of DNA. The egg has half the normal amount of DNA, and when you combine them, we have 100% the normal amount of DNA. Okay, let's take a look at this diagram on the left here. Starting up at the top here, we have interphase. So that would be G1, S, and G2. And remember that in S phase, we have replication of the chromosome. So over here, I'm gonna ignore this 2N for now. I'll explain that in a couple of slides later. What we have is 1, 2, 3, 4 chromosomes, and each one is just represented by sort of a line, a thick line. And what happens before meiosis occurs, just like in mitosis, is the DNA is replicated. So notice the difference between A and B here. B, the chromosomes are represented as sort of like an X, and then in A, they're just a line. So DNA replication takes this one chromosome, copies it, and then its copied self is joined to itself at the centromere. And we call these sister chromatids. Okay. Next thing, and this is, I'm going to skip through it really fast. We'll go into the details later. Beginning at the start, again, A, we had 1, 2, 3, 4 chromosomes. 4, right? You can see 2 blue, 2 red. And then we copied the DNA, so now there are sister chromatids. And then there's a nuclear division, and we get to cell C here. Now we have 2 cells, right? So 2 cell Cs, and each of them only has 1, 2 chromosomes, each with their sister chromatid. This one also 1, 2 chromosomes, each with their sister chromatid. So we divided the DNA by half, divided by 2, and we get this. So if you divide B by 2, then you get these 2 cells down here, C. Next, we're going to undergo a second nuclear division, and in this case, it is pretty much the same thing as mitosis, where we've got these sister chromatids joined by the centromere, and we're going to divide them down the middle, like so. And what we get is these cells down here, D, 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 D. And so you can see that this cell, when it divides, each of these is divided right down the middle, and we get 2 cells, each with half the amount. Now, let's look at back at our initial description here. Let's see, reduces the chromosome number by half. Okay, so in cell A here, there are 4 chromosomes. In cell D, there are only 2. So we went from 4 to 2 chromosomes. Oh, one more thing I should mention about meiosis, something you want to remember is, we'll say bullet 5, is there are 4 daughter cells. There's 4 daughter cells. So one cell becomes 4 daughter cells. All right, so sexual reproduction, involves 2 parents. And so let's imagine that we have some kind of organism that only has 2 chromosomes. So here's a cell. This cell, let's say it's a nucleus. Okay, this is the nucleus of some cell, and it just has 2 chromosomes. It got this chromosome from mom. So we're going to say that, now we're going to use the science word, this is the maternal chromosome. And then we'll say this chromosome down here, it got from dad, and the science word is paternal. The paternal chromosome. Another thing is we're saying that these are homologous chromosomes. This is a homologous pair of chromosomes. What homologous means is, well, H-O-M-O, that's a root word that means same. So what that means is same genes. Same genes. Just like you know that you have gotten some of your eye color genes from mom and some of your eye color genes from dad. Both dad and mom contribute a copy of every single gene. And here's why that is. For every chromosome that you have, and in this imaginary cell, this cell just has a single pair of chromosomes, but humans have 23 pairs total, so 23 pairs, so 23 from mom, 23 from dad, equaling a total of 46. And each of them is the same as the other. So the ones that you get from mom are the same as the ones that you get from dad. They have all of the same genes. So what can we say here about this very simple example? This we could call a gene, okay, and it's a gene about color, purple flowers, all right? Down here, we could call this a gene, and it is also a gene for the color of flowers. Now, how are they different? I said that you get all of the same genes on the maternal chromosome, all of the same genes from the paternal chromosome. Well, you get one thing that is different, and that is the alleles. So what is an allele? An allele is a version of a gene. It's a version. So we said that this is the gene for flower color, and this version is purple. Down here, we said this is the, right here, this is also the gene for flower color, and the version of this gene is white. So this individual from the paternal side got the white version of this gene, and from the maternal side got the version of the purple, the purple version of the gene. So alleles are just different versions of genes. More on homologous chromosomes. So again, we have a homologous pair of chromosomes. Let's take a look at them and note that we can see a couple things just by looking. Number one, they are always the same size. Another thing, okay, not from looking, is that they have 100% the same genes, but they do have different alleles. So they may have different versions of a gene, right? So we had maybe the purple color here and then the white color here, different versions of the same genes. Next, when a cell, or a nucleus rather, is beginning to divide, before that in S phase, the chromosomes will be duplicated. So this one, the paternal chromosome will be duplicated, the maternal chromosome will also be duplicated. So sometimes these same chromosomes will be shown like this. And all that means is that replication has occurred. This red maternal chromosome has been copied, and now there's two copies of it. The two copies are joined at their centromere. So there's the centromere. Same thing with the paternal chromosome down here. This orange chromosome has been copied, and its copy is joined to the original at its centromere, which is right here. Centromere. I'm not going to write it again. Another thing about homologous chromosomes. How do we identify, under the microscope, for example, that two chromosomes are homologous? Another thing is that the centromere, oh, here I am writing it again. Centromere is in same location. So notice that it's right about in the middle here, it's right about in the middle here, but it's maybe a little bit more towards the top, or it's a little bit more towards one end of the chromosome. Another thing that we can use to determine if two chromosomes are homologous is we can look at the banding patterns, and that is essentially we can stain chromosomes. Remember, light microscopy can use stains to better clarify what we're looking at, and essentially we get these kind of black and gray and white banding patterns. And homologous chromosomes will always have the same banding patterns, like so and like so. Then this one would be exactly the same. 1, 2, 3. Is that 3? Okay, yeah. So this is another way that under the microscope we can match them up. And for example, if we saw a cell that had, say, maybe a third copy of a homologous chromosome, we would know that something is wrong there. Or conversely, if a cell was missing one of the chromosomes, because in human beings we should have two copies of every chromosome, and if we were missing one, that would also indicate that there is a problem with the cell, the tissue, or even the whole organism. Next topic is ploidy, and this refers to the number of sets of chromosomes. Number of sets of chromosomes. In a haploid cell or a haploid organism, we would say that the organism has one set of chromosomes. We can also say n. It has n sets of chromosomes. Over here, in a diploid organism, all the cells have two sets of chromosomes, and again refers to one set, so 2n sets. And we can do a little bit of math here to say, well, if over here n equals 3, that means there's 3 chromosomes, 1, 2, 3, then there is 3 chromosomes in each cell. In a diploid cell, 2n equals 6. There are 6 chromosomes in each cell. Now, a diploid organism, such as a human being, a lot of plants, most animals, are generally, not always, but generally the result of, let me write this better, sexual reproduction. Sexual reproduction, meaning that, for example, this pair of chromosomes, this is a homologous pair. Notice the centromeres are in exactly the same place, and one of the chromosomes, we'll call the one on the left, came from mom. We call it the maternal chromosome. The other chromosome came from dad. We call it the paternal chromosome. Same thing for the remaining two homologous pairs. This homologous pair, the centromeres, are of course in the same location. One is from mom, maternal, one's from dad, paternal, and then the same thing for this last one. Maternal, paternal, oops, that almost turned into a B. So it's supposed to be a paternal. There we go. So diploidy, number of sets of chromosomes. And let me make a distinction here. Down here, what we have is, let's see, how many chromosomes are there sitting in this cell? Well, we would actually just say that there's 1, 2, 3, 4. Now, why is it that I'm just saying 1, 2, 3, 4 and not maybe 1, 2, 3, 4, 5, 6, 7, 8? Why am I not saying that? The reason is that this is one chromosome, each, and it has its sister chromatid. So what we can say is this one, well, this whole cell here, DNA has been copied, and each chromosome is bound to itself by its centromere there. And then this one's kind of a funny looking chromosome, isn't it? And also bound by their centromere. In this case, we would say, how many homologous chromosomes are there down here? Well, this would be one pair of homologous chromosomes. And again, notice that the centromere, right here and here, they're in the same place, yeah? So this is one homologous pair there, and then we've got another homologous pair there, so two homologous pairs. So we'd say 1, 2, 3, 4. This one could be the maternal, this would be the paternal, this would be the maternal, and this could be the paternal. And we could reverse this. I mean, if that's confusing to you, I could call this the paternal and this the maternal. That's just random. Okay, so ploidy, again, number of sets of chromosomes. Here, one set each. One chromosome. Just one there too, just one hair, right? One set of each. There's 3 different chromosomes. 1, 2, 3. Over here, diploid, we've got 2 copies. 1, and then the second copy. So we've got 2 copies of diploid. All right. So meiosis 1. Remember that meiosis involves 2 nuclear divisions. So this is the first nuclear division. The first nuclear division. So in interphase, what we're going to have happen is during S phase, there will be DNA synthesis. We therefore get chromosomes in prophase 1 that look like X's, like so. Another thing that happens is you can see that they are sort of paired up. So there's sort of a blue X and then a red X there, right? So blue, there's a blue one and then there's an X. Sorry, let me change the color. Working on it. Okay, I got it. And then there's another one there. So what I'm drawing there is exactly what is shown here, but it's a little bit fuzzy. So we have for example, maybe a maternal and then a paternal chromosome. Each of them, their DNA has been replicated, and they now pair up. And that becomes important when we go to the next phase, which is metaphase. Just like in mitosis, sort of. They do line up at the equator, but what is lining up at the equator? In this case, we have homologous pairs lining up. Homologous pairs line up. Does it say that there? No. Homologous pairs line up here. And we can also say that these are called a tetrad. We'll come back to that later, but this is called a tetrad. Here's a tetrad. Here's another tetrad. It kind of makes sense. Tetra means 4, and there is sort of a total of 4 copies in a way. So the homologous pairs line up, and then in anaphase 1, similar to mitosis, they're pulled apart. But in this case, the sister chromatids remain joined. So here what's going on is both maternal chromosomes, the red ones in this case, are going to the left, and going to the right, both paternal chromosomes are going to the right. So we have division of the chromosomes. In telophase, briefly the nuclear membrane forms, cleavage furrow develops, cytokinesis occurs, and we get two cells. Each of these two cells will then divide to form another two cells for a total of 4 cells. Next, taking these two cells, moving on to the next slide. So we'll look at these exact same cells and look at the next steps of meiosis. The next steps of meiosis, again, those two cells here, is the second division. The second division. So we have started off, remember that we started off with all of these chromosomes in these two cells in a single cell. So the single cell that gave rise to this two chromosomes would have looked, as a reminder, you could just go back on back and look at the last slide, would have looked like, okay, there's that chromosome, then there's the kind of bigger red one, and then there's the same homologous chromosome from the paternal side, and then there's a littler blue one as well. So cell A became cells, cell 1 and 2, like so, divided in half. Okay, so now what happens is very, very similar to what happens in mitosis. We go to, this is prophase, not a whole lot of interest to describe here in prophase. What happens though is in metaphase, the chromosomes line up at the equator, and just like in mitosis, very similar to mitosis, but then in anaphase too, the sister chromatids divide, just like mitosis. The centromeres are split, and the chromosomes move to opposite ends of the cell, or opposite poles of the cell. Telophase, nothing much to describe here that is different than mitosis, and we get 4 haploid cells. Let me make one final comment here. Look at these 4 cells. They are not identical. They're not identical. And this cell, we'll call this one cell A. Cell A has one big blue one and then a little red one. And then cell B has one big red one, then a little blue one. And then cell C down here has a slightly different arrangement. And also notice this. This chromosome here and then some of the other ones are sort of a combination of red and blue. And we haven't talked about that yet. That is called crossing over, and involves the exchange of DNA from one chromosome to its homologous partner on the maternal or paternal side. We'll get to that. Crossing over, how DNA is exchanged from one chromosome to another. The final result of this is that there is a recombination of alleles. So let's take a look at the top here. Prophase 1 of meiosis, we have non-sister chromatodes held together during synapsis. You can look at what synapsis is in your book. It is essentially the binding of chromosomes to each other, homologous chromosomes to each other. So they are bound to each other here, and it's also shown right here, bound to each other. And essentially what happens is they get tangled up, and after they have been tangled up, and I shouldn't say tangled in that it is random or bad, they associate with each other, and something called chiasmata form. These chiasmata bind chromosomes that are not sister chromatids to each other, and then the DNA is cut, and then it is recombined with its non-sister homologue. So you can see down here that the very tips of the blue one and then of the red one have been exchanged, right? So this one has been exchanged for this one. The DNA exchanged, it is the same amount. So it's the same amount of DNA, same amount of DNA. So if we take the top 5% of this chromosome and trade it for some amount of the other one, it's going to be the same amount. So the top 5% was exchanged and for the other top 5%. The result is shown here. This is the result of recombination. So we can see that this, if we're going to say, let's say blue is paternal and red is maternal, what we can see is that this chromosome that is mostly a maternal chromosome now has parts from the paternal chromosomes. In this case, it looks like 2 crossover events occur. Over here, the chromosome that is mostly blue or mostly paternal has received some of the maternal chromosomes. So this basically shuffles alleles around. This shuffles alleles around. And we'll look at the consequences of that in a later chapter. What we can say for now, though, is this. Let me just create a little bit more space for myself here. One of the key features of meiosis is that genetic variation results. The daughter cells are not identical to each other. Notice that cells 1, 2, 3, and 4, none of them are identical to each other, even when we look at just a single chromosome. Cell number 1 has one paternal chromosome where crossing over has not occurred. Cell 2 has a mostly paternal chromosome where crossing over has occurred. And so the alleles that are on this chromosome are slightly different. Remember that when we started off up here at the top, we had 2 paternal chromosomes bound to each other in some kind of fashion like this. And that they would have been 100% identical because they're the result of DNA synthesis. However, because of crossing over, parts of the paternal chromosome were exchanged for the maternal chromosome. Therefore, the alleles present now on each of these 2 chromosomes are different. And then the same thing goes for cell 3 and cell 4. Cell 4 has just one 100% maternal chromosome, and then cell number 3's got a mostly maternal, but some of the small regions of it have been replaced with paternal sequences of DNA. Next, independent assortment. So here we go, independent assortment. Let's take a look at, let's see, this would be metaphase 1, oh, it's written right there, metaphase 1, and the homologous pairs have lined up at the metaphase plate or the equator of the cell. Both of those are correct. And we could have one possibility where all of the paternal chromosomes, the blue ones, are on the left side and all the maternal ones are on the right side. That's possible. Possibility 2 is that it's more of a 50-50 split in this simple cell, where one paternal chromosome is on the left and then the other paternal chromosome is on the right. This is why we say that this is why we use the word independent assortment. What does that mean? It means that the orientation of the chromosomes, they are independent of one another. So let me clear up some of this here. The orientation of this big chromosome on the top, whether it is on the left or the right, is independent of where this chromosome is on the bottom. So and literally 50% of the time they'll be on the same side, and then over here 50% of the time they will be on different sides of the equator of the cell. The result is important. Possibility 1, anaphase occurs. Everything on the right goes to one cell, everything on the left goes to another cell, as shown here and here. metaphase 2, and then also meiosis 2. We're simplifying it. We're not discussing anaphase and all that because we've already mentioned it quite a few times. What happens now is these sister chromatids divide, and then we get these daughter cells and these daughter cells. In this example, no crossing over has occurred. But what the result of the point being here is, even though no crossing over has occurred, the fact that these chromosomes assort independently into the daughter cells means that there is more variation in cell type. So this independent assortment produces variation. So no crossing over has occurred. So these two cells, yep, they're 100% the same. These two, 100% the same. So we're calling them combination 1 and 2. Then over here, combination 3, yes, these two are the same. And then combination 4, these two are the same. And how I'm saying they're the same is just, well, how many big red ones and how many little red ones and little blue ones and big blue ones. Combination 4, all the same. Combination 3, all the same. So variation, in what way is the variation produced? The variation is produced in what combination or combo of paternal and maternal chromosomes are inherited. What combination of paternal and maternal chromosomes are inherited? Combo 1 is all paternal, combo 2 is all maternal, combo 3 is a half and half, and combo 4 is sort of a different half and half. So again, bottom line is independent assortment, or the arrangement of the chromosomes during metaphase, increases the amount of variation produced in the daughter cells. Here is an animation of meiosis, and it does sort of start off with a very zoomed out view, and then it zooms in just to give you a context of where this is happening.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, so that is pretty much wraps up meiosis. However, I'm going to go into greater detail about meiosis and the movement and behavior of chromosomes during our lab lecture. So if that was a little bit confusing to you, what I would say is that that is absolutely to be expected. What I would do is review the material in your book, and then we'll also get into more details of it in the lab lecture and lab activity. Okay, so this slide is about comparing mitosis and meiosis, and then there is a description of the two at the bottom, and I would absolutely read every single item on here. I am not going to read through it for you though. I'm going to talk about the diagrams up here. Okay, so let's see. Beginning with prophase in mitosis, what happens in prophase is pretty much the same thing in meiosis, in that DNA replication has occurred. So we can see the chromosomes sort of look like X's. So that means DNA replication's occurred, joined at the centromere. But in meiosis, what happens is, yes, the chromosomes have been replicated, but also homologous chromosomes pair up like this, forming a tetrad. That's called a tetrad. It just makes it easier to refer to. What is this? That's a tetrad. Maternal, paternal. And crossing over can occur, and then we move to metaphase. And in metaphase, in mitosis, we just have one chromosome, each with its sister chromatid. So like this, yeah? But over here in meiosis, we have 2 chromosomes lined up, homologous pairs, maternal and paternal. Anaphase in mitosis involves the centromere dividing. Anaphase in meiosis 1 does not involve the centromere dividing. Notice we still have these X formations because in anaphase, each homologous pair moved to the left or to the right depending on what side of the equator it was on. So now, where was I here? So let's see. In mitosis, the division is complete. So mitosis is comparatively very simple. We essentially have the synthesis of chromosomes, then they line up like an X, like that, right? And then the centromeres divide, and then we get one piece on either side, or one chromosome on either side. But in meiosis, the second division here, this is meiosis 2, and it's condensed into just a single sort of cell here. What happens is, again, we have all of those in a metaphase. They all line up like so, and then they divide similarly to mitosis, producing 4 daughter cells that also just have 1, 2, 3 chromosomes, half the number of the parent cells. So let's see, in mitosis, right here, let's review what we could say about ploidy for mitosis and then ploidy for mitosis as well on the right here. Let me just give myself some room to write. Okay, so we can just count the chromosomes in mitosis. 1, 2, 3, 4, 5, 6. So we have 6 chromosomes. 6 chromosomes. That means we have 3 homologous pairs. We have 3 homologous pairs. So that would mean that we have, let's see, n sets of chromosomes. Human beings, if this was in a human being, are diploids. So let's see, 2n. 2n, n is 3, because there are 3 homologous pairs. There are 3 sets, so 2n equals 6. Let's look over at meiosis again, and we can see, let's see, these two are a homologous pair, but so we have 2 chromosomes. These 2 chromosomes are a pair. Then 3, 4, these 2 chromosomes are a pair. 5, 6, these 2 chromosomes are a pair. So again, we've got 1, 2, 3, 4, 5, 6, and we have 2 sets of chromosomes. How many chromosomes is one set? One set is 3 chromosomes. So we say n equals 3, and this cell is diploid, so 2n equals 6. Review this in your book now if you're still a little confused on what ploidy is. Let's go down here. The daughter cells here, there's 4 of them, and each one is n. Notice, daughter cells in meiosis 2, n. 1, 2, 3 chromosomes in each. So they each have 1 set of chromosomes. So n equals 3, or 1 set. In mitosis, what happens is we have doubled the amount of DNA by copying it, DNA synthesis, and then we line it all up in the middle, and half goes to the left, half goes to the right. So because we doubled it, then divided it, we are right back at where we started, 2n. We can count the chromosomes again. 1, 2, 3, 4, 5, 6. So 6, and if we arrange them by size, we notice that there are 2 sets based on size, location of centromere, and banding patterns. We can't see the banding patterns here. 2 sets, n equals 3. They are 2n. 2n equals 6. We counted them up. 1, 2, 3, 4, 5, 6. Okay, summary of meiosis. Yes, DNA was doubled by DNA synthesis, but then we had 2 rounds of division. So if you double something and then divide it 2 times, you get to 1/2 the normal amount. Please make sure that you read this down here before moving on. Here is an example of the aphid life cycle. I mentioned that most animals reproduce sexually. Insects are often an exception. They will do both, and they do it in a whole wide variety of ways, strategies. If you're a gardener, you know that aphids are bad for your plants. Here's why. They hatch in the spring on a host plant, and they are all wingless females. By the way, you don't need to memorize this. This is purely food for thought to help you kind of connect some ideas here. Aphid life cycle. They hatch in the spring from eggs, producing larvae. They're all wingless females, 100% females. The wingless females reproduce asexually, producing 5 to 10 clones of themselves every day. And they're all female, so 5 to 10. So a single female at a maximum. Day 1, there's 10. Day 2, all 10 of those, well, it takes them a little while to mature, but each of those 10 females will produce 10 more at maximum. So now we're up to 100, and then 1,000. So super fast reproduction. After host plant is devoured, they grow wings and fly to another plant, continuing to reproduce sexually. So you can think of some type of cell signaling or environmental signaling model going on here that causes them to grow wings. Perhaps a shortage of nutrition causes them to grow wings. In the fall, temperature decreases, which transforms some of the females into males. Sexual reproduction commences, generating genetic diversity. In warmer climates, sometimes no males are generated. That depends on the species. There are hundreds of species of aphids. So temperature decreases, causing them to change their biological sex. So again, some type of cell signaling, probably causing gene activation or gene inhibition is going on here. Sexual reproduction, why are we doing that? Because that produces genetic diversity, which means that they're going to be more resistant to predators. They may be more resistant to diseases, and they may also be more resistant to plant toxins that the plant is producing to hopefully try to stop the insect from eating them. So the idea here is that asexual reproduction means we can have a population explosion really fast. Sexual reproduction, that means that we can generate diversity so that the offspring are suited to a variety of environmental conditions, including what kind of plant are they eating, what kind of diseases are present, etc. The human life cycle. So this is the last topic in your book, and it is a little bit confusing, perhaps. Consider this. We generally think of human beings, our whole life, we spend as creatures with 2 arms and 2 legs, a head, feet, all those things, right? Essentially, we can say that the dominant phase of our life cycle is 2N, or diploid. When we produce sex cells, so sperm or egg, by meiosis, we now have half the amount of DNA in each of these cells. So is this part of being a human being right here? Is being an egg part of your life? Was being a sperm part of your life? Well, we say that, well, probably not. Your answer to that would probably be no. So what we'd say is that this down here, dominant phase of our life cycle. But imagine if this were reversed, perhaps producing a diploid zygote. It grows into a human being. The human being grows only enough to be able to produce more sperm and more egg cells. And these humans, let's just imagine that they are in fact very low intelligence and they can't really do anything other than reproduce. The sperm and egg cells, however, are geniuses. They go off to university, wherever egg cells and sperm cells go to university, and they are the dominant phase of human beings. This is clearly not true. Okay, can you imagine an alien species that is just so bizarre that how they view life is this temporary and inconvenient thing called a body, or a macro stage of life, or the multicellular diploid stage. These things are purely a means of producing eggs and sperm, which are clearly the more important part of life. That could be possible in an alien species. What about this? Is it possible here on Earth? And the answer is yes. All right, let's first consider the bird, which is pretty similar to human beings or mammals, and let's look at its life cycle. So a bird is produced when gametes, the sperm plus an egg cell are always bigger. The sperm fertilizes the egg, and we get N chromosomes from the sperm, N chromosomes from the egg, to get 2 N chromosomes. And this zygote is going to divide by mitosis, producing a multicellular diploid organism. And this is most of the life cycle. This constitutes most of the life of the bird. The bird will eventually have sex cells once it is sexually mature, but undergo meiosis, and these cells, the results of meiosis, they immediately produce gametes. Immediately produce gametes. And you might say, well, of course they immediately produce gametes. What else would they do? Gametes are for sex. Now, gametes in birds and human beings are haploid, like as we've said, just N sets, right? 1 copy. But that's not true for all organisms. This will make sense if we now look at this unicellular organism over here. Many protists are like this. Let's consider what's going on here. This cell right here, it is haploid, unicellular or multicellular haploid organism. What is it doing? It is swimming around, and it is swimming around doing all of the things that living things do. Getting food, responding to stimuli, and eventually reproducing. Sometimes, so this is haploid right now, yeah? Sometimes gametes can be produced by mitosis. So what that means is that a single cell that is N is going to divide by mitosis to produce gametes. So mitosis produces gametes. Just as in, so what that essentially means is that one cell type can divide to produce different cell types. So this is the sort of adult phase of the organism, and the entire, because it's just one cell, the entire organism divides producing only sex cells, two sex cells specifically. These two sex cells are then going to participate in sexual reproduction with different sex cells from a different unicellular organism. So these two N plus N sex cells, they are going to fertilize one another, producing a zygote. Now that is 2N. The thing is here is that the zygote is now not going to do mitosis. No mitosis. It's not going to grow and develop into a blastula, an embryo, a fetus, and all that. What it's going to do is it's going to immediately undergo meiosis to produce more haploid cells. These haploid cells go about doing all the things that this creature does, swimming around with its flagella, gathering food, running away from predators, all that stuff. So why is it that this organism even bothers to produce these gametes that then fertilize one another, producing a very short-lived zygote that then produces more haploid single-cell organisms? Why? Because of one of the features of meiosis. Meiosis increases variation in offspring. So this is evolution's way of hedging one's bets. So rather than having this single cell here just having one genetically identical, let me say that again, rather than this single cell just dividing to reproduce by mitosis and producing genetically identical offspring, what it does is it produces those gametes that undergo meiosis, and that increases the variation in its offspring, because meiosis involves crossing over, because we can have more than one parent cell, this cell and this cell, from two different cell lineages, and we can recombine genetic material, hoping that one of these combinations or many of these combinations will be well suited to the environment. Okay, meiosis increases genetic variation. So last one here, down here. So this is plants, some algae, some fungi. They have a similar sort of situation, but it is more balanced. So the gametophyte is a multi-celled, that says multi-celled haploid organism. So each cell has N sets of chromosomes, and these undergo mitosis, and that's how they grow. That's how, let's see, this spore that was N, right? It undergoes mitosis and produces a multicellular organism that's big enough for you to see with the naked eye. And this haploid organism, N sets of chromosomes, goes about doing what it is that plants do. They do some photosynthesis, they grow these little structures down here to grab onto the rocks that they're growing on, or whatever it is they're growing on. They live their life and eventually will produce gametes by mitosis. So mitosis produces gametes, and these gametes are cells that are involved in sexual reproduction. So now we can have two parents, N plus N. So we can have the combination of genetic material from this parent and this parent to form a zygote that is now 2N. This creature, happening to be a fern, is going to undergo mitosis similarly to a human. So zygote, blastula, embryo, fetus, etc. except it's a plant. It doesn't have those exact structures. It is going to grow by mitosis, producing more cells, different tissue types, different cell types, and it's going to do what plants do: photosynthesize, absorb water, etc., etc. Eventually, when some environmental stimuli comes along, it's going to undergo meiosis, which is going to produce haploid cells, N, but they are not gametes. So gametes, remember, are sex cells. Gametes are sex cells. These are simply haploid cells. And these haploid cells undergo mitosis to produce a multicellular organism. So the idea here is alternation of generations, meaning, and the Fern perhaps characterizes it the best, is it alternates between being haploid and between being diploid. Both phases of its life cycle are somewhat equivalent in terms of multicellularity and duration of lifetime spent in the N phase versus the 2N phase. Vertebrates like birds and mammals, it's pretty unequal. Most of the time we are in our diploid phase. We only do meiosis to produce sex cells, gametes for sex, and then immediately go back to our diploid phase. That is alternation of generations. I spent a little more time talking on that last slide, so I'm not going to talk too much about this. This is just another example. This is moss, and there are multiple structures here. The sporophyte here, this is 2N. All the brown colored items here, this fella here, and you can see this on moss. Next time you see moss, you can look at that. And this is the only 2N part, and it grows out of, let's use a different color, this thing, which is N, haploid. So haploid, that's the gametophyte. You don't need to memorize the term gametophyte. You just need the general idea that there are different life stages. Some of them are haploid, some of them are diploid. So this haploid multicelled structure here produces a diploid structure because these parts of it can be fertilized, and then this thing grows into a spore-producing thing, which undergoes meiosis, producing haploid cells that then develop into a multi-celled organism. So again, this whole blue section here, this is the haploid stage of this creature's life cycle, moss. So what is meiosis anyway? You can discuss these questions on your discussion board if any of them seem to be very difficult for you. You should be able to answer all of them, not in terms of regurgitating what I've stated, but in terms of thinking about what I've talked about and answering these questions. And then these last four are about this question: what phase are each of these in? And this one I'll help you out with. So what phase is this of meiosis? This would be metaphase 2. I know that because the sister chromatids are lined up at the equator, not homologous pairs of chromosomes. This one would be prophase 1. I know that because there are homologous pairs present. This is a tetrad here. This is another tetrad, kind of hard to see. And it looks like crossing over is happening as well. You can see that. Looks like it's going on right there. And then this is going to be anaphase 1. And I know it's the first anaphase because what's getting pulled to opposite ends of the cell is a chromosome with its sister chromatid still attached. In anaphase 2, it's different. And then here is metaphase 1. And I know that because these tetrads are lined up at the metaphase plate. That means homologous chromosomes, each with their sister chromatid attached. This is a karyotype, and these can be created by, again, staining. So you can see those banding patterns right here, and homologous chromosomes have the same banding patterns. You can see. What we can see is that this individual has gotten 3 copies of chromosome number 21, and this is in fact called trisomy 21 and is one of the most common forms of Down syndrome. Down syndrome. Sometimes doctors, and I think this may be a little bit outdated now, but if they suspect that a fetus has Down syndrome, then they may take a sample of its cells and create this diagram here, a karyotype, by staining its chromosomes and then from one cell and then lining them up like so. And essentially what has happened here is that during either anaphase 1 or anaphase 2, and remember all these chromosomes line up at the metaphase plate, there has been an unequal division of cells. So maybe in anaphase 1, let's say they divided correctly to produce one cell's got that and the other cell's got this. Then in anaphase 2, let's take this example. This chromosome is lined up at the metaphase plate, and let's say this is chromosome number 21. And we know those microtubules are going to grow from these centrioles, right? Grab the kinetochore. What if the centriole on this side, centrosome, each of these two things are centrioles, what if this one never quite grabbed the kinetochore? Well, what that would mean is that this whole chromosome with two sister chromatids would go to this cell, and this would result in a gamete that has two chromosome number 21. So if you had a sperm cell that had two chromosome 21s in it, it's got two of them, then remember the egg cell is going to have another one chromosome number 21. So 2 plus 1 is going to equal 3 chromosome number 21s. So this can be used to diagnose Down syndrome. And that is all. So we'll talk more in detail about the behavior of chromosomes during our lab on meiosis.